Hello everyone. In today's video we will learn about the biochemical basis of the changes seen in diabetic ketoacidosis. So today's topic would be biochemical basis of the changes seen in diabetic ketoacidosis. So we'll look into the detail about all the cause of laboratory findings which we see in diabetic ketoacidosis. So the first being hyperglycemia. So why there is hyperglycemia, we'll look into that through a flow chart so that everybody can understand it in an easy manner. Okay, so the first finding which we see, laboratory finding in diabetic ketoacidosis, is hyperglycemia. That is increased blood glucose level. Okay, so what is the basis of this hyperglycemia? So all of you might be knowing this, in diabetes individual who have this acute complication called diabetic ketoacidosis, insulin level is almost absent. Okay, there is a lack of insulin. So there is lack of insulin. So what will happen? There will be decreased utilization of glucose. Decreased utilization of glucose by peripheral tissues. Like peripheral tissues, like what? Like skeletal muscle, adipocytes, as this GLUT4 mediated uptake, of glucose is reduced. Since this GLUT4 require insulin for its activation. Okay, so there will be GLUT4 will be we won't be active due to deficiency of insulin in diabetic ketoacidosis patient, and because of that the glucose won't be going inside tissues like adipose tissue, skeletal muscle or cardiac tissue. So that will lead to what? Hyperglycemia. So this is the main cause of hyperglycemia seen in diabetic ketoacidosis. Apart from that, the other reason being, since the tissues are not able to utilize glucose properly, so what will happen? There will be increased catabolism. Okay, so this diabetic ketoacidosis is highly catabolic state, okay, where there is a breakdown of proteins, especially that muscle protein breakdown will be very high. So this muscle protein breakdown will release one amino acid called alanine, and this alanine will go inside liver, where this can be utilized to produce glucose. So there will be increased gluconeogenesis inside hepatocyte. So liver will synthesize more and more amount of glucose, and this glucose will be added up into blood. So already there is a decreased utilization of glucose by peripheral tissues, and on top of that liver is synthesizing more and more amount of glucose through non-carbohydrate sources. So that will further add on to hyperglycemia. So that is the basis of hyperglycemia in diabetic ketoacidosis patients. So the point here there are two main points to remember. The first point being, there is a decreased utilization of glucose by peripheral tissues like skeletal muscle, cardiac tissue or adipocyte due to GLUT4 inactivation, because GLUT4 is not working properly, and GLUT4 is a insulin dependent glucose transporter which requires insulin for its activation, and for uptake of glucose. And this GLUT4, remember, it is expressed mainly by cells like adipose tissue, skeletal muscle or cardiac tissue. So obviously that will lead to decreased utilization of glucose by these tissues due to lack of or inactivation of GLUT4, which requires insulin. So we don't have enough insulin, so GLUT4 won't get activated, and tissues like adipose tissue, cardiac tissue or skeletal muscle won't be able to take up glucose inside it, and they won't be able to utilize glucose. So obviously there will be build-up of glucose inside blood, and that will lead to hyperglycemia. Second point is, liver try to synthesize more amount of glucose by using non-carbohydrate sources like like protein. Muscle protein will be broken down, and there will be excess flow of alanine inside blood, and this alanine will go inside liver, and that can be used to produce glucose. So there will be excess buildup of glucose inside liver, and that glucose will go from liver into blood. So that would further add on to hyperglycemia. So these are the two main causes of hyperglycemia seen in diabetic ketoacidosis patients. So that you should remember. The second finding is there is increased formation of ketone bodies. Ketone bodies. So there is, we call it as ketosis. As the name suggests, you can see diabetic ketoacidosis. So obviously there is hyperglycemia, and ketoacidosis means due to increased production of ketone bodies or due to increased level of ketone bodies in blood, that pH will go down, pH of plasma will go down. So that will lead to what? Acidosis. That's why that name diabetic ketoacidosis has come from. Okay. So why there is increased formation of ketone body? As there is lack of insulin is low. So the counteracting hormones like glucagon level will be on higher side. So this glucagon will stimulate lipolysis. So this glucagon is going to break down lipid in excess amount or fat in excess amount. So what will happen? This glucagon will stimulate breakdown of triacylglycerol, that is a storage form of fat, into free fatty acid and glycerol. Okay, and this free fatty acid will be released into blood. Once it is released into blood, it will be taken up, or it is going to bind with albumin. So there will be formation of albumin free fatty acid complex. And this albumin and free fatty acid will be going into liver. Okay, and inside liver, this free fatty acid will be broken down into acetyl coenzyme A. Acetyl coenzyme A. So there is increased free fatty acid, and of course there will be increased level of acetyl coenzyme A. So what will happen? This excess amount of acetyl coenzyme A will be diverted into synthesis of ketone bodies. So these ketone bodies are what? There are basically three types of ketone bodies. So there will be increased level of acetone. That's why there is in breath we can smell. There will be smell of acetone in person's breath because it has a fruity odor. So there will be increased level of acetone. There will be increased level of acetate, and beta hydroxy butyrate. So these all are ketone bodies. So there will be increased formation of all these three ketone bodies by liver. So what will happen? So liver will send those ketone bodies which are synthesized in extra amount into blood, and in the blood the level of all these three ketone bodies like acetone, acetoacetate and beta hydroxy butyrate will be very, very high. And of course the peripheral tissues won't be able to utilize all these ketone bodies. So the level of ketone bodies in blood will increase, and that will lead to a condition called ketoanemia. So there is a ketoanemia, that is the excess level of ketone bodies in blood. So there is condition called ketoanemia. And once that ketoanemia happens, of course after a while that ketone bodies will start appearing in urine. So that is called ketoanemia. ketonuria. So in diabetic ketoacidosis patient, if we see there will be increased formation of ketone bodies. There will be increased excretion of ketone bodies in urine, and that is called ketonuria. And ketonemia plus ketonuria combined together, these two things together, that is called ketonemia and ketonuria together is called ketosis. So what will happen if the ketone bodies level in blood increases, and these ketone bodies are acids? Okay, so they are acids means what? They will secrete H plus ion, so there will be excess secretion of H plus ion. This H plus ion is going to reduce the alkali reserve of our body, because there will be this bicarbonate, which is a major buffer system in our body, and that ensures that our blood pH remains around 7.4. So this bicarbonate will ensure, so we'll try to due to that excess H plus ion formation, and because of that ketone body which is secreting more amount of H plus ion, this bicarbonate will be used up to neutralize the effect of H plus ion. So the bicarbonate level will also go down, and of course at one point of time when all the bicarbonate is exhausted or bicarbonate level is on the lower side, it won't be able to restore the pH. Okay, and our pH will go down, so there will be decrease in pH, there will be decrease in bicarbonate level due to excess formation of ketone bodies, and that will lead to a condition called metabolic acidosis. Okay, so this is what we see in diabetic ketoacidosis patient due to decreased bicarbonate, and of course due to formation of excess H plus ion by these ketone bodies, our pH and our bicarbonate level will go down. So that's why there is a metabolic acidosis, and the compensatory mechanism for that, the counterbalance that effect of change in pH is also there will be decrease in PCO2. So this is the secondary change. Through hyperventilation, our body system will try to defense system will try to restore the pH to 7.4. So in order to restore that pH, there will be hyperventilation, and there will be expulsion of carbon dioxide through lungs. So there will be decrease in PCO2 also. So if you see in diabetic ketoacidosis, what pattern which we see? There is hyperglycemia, there is ketosis, there is decrease in pH, there is decrease in bicarbonate. So all those metabolic changes cause, we have seen the basic concept. Now if you see in diabetic ketoacidosis, there is hyponatremia, mild hyponatremia. Hyponatremia is reduced sodium level or mild decrease in sodium level is seen. Why that happens? Because glucose level will be on higher side, and this glucose level in blood will be high, so that will have that osmotic effect. Okay, it's going to pull water from, this glucose is going to pull water from ICF to ECF compartment, and that will cause dilutional hyponatremia. So in diabetic ketoacidosis patient, if we see that sodium level will be slightly on the lower side. So that is one of the finding, that is due to that osmolality of that glucose, which is very high inside the blood, and this glucose will pull water from ICF to ECF compartment, and that will cause dilutional hyponatremia. That is a mild decrease, there will be mild decrease in sodium level. And in diabetic ketoacidosis patient, there is also hyperkalemia. Hyperkalemia nazar aata hai. That is due to what? There will be increased level of potassium in blood. Why that is happening? Because that there is a deficiency of insulin. So insulin is lacking here. So this insulin is responsible for shifting of that potassium from K plus from ECF to ICF. So this insulin is responsible for shifting of potassium from ECF compartment to ICF compartment. So it promotes intracellular trapping of potassium. So here that intracellular trapping is due to deficiency of insulin is not taking place, so that there will be buildup of potassium inside ECM, and that can cause what? Hyperkalemia, that is increased level of potassium in serum. So that's what we see in case of diabetic ketoacidosis, and these are the major findings which we associate. So there is hyperglycemia, there is there is a ketosis, there is due to ketoacidosis there will be a decrease in pH, decrease in bicarbonate, so that will lead to metabolic acidosis, and there will be apart from that there will be hyponatremia due to osmotic effect of increased level of glucose in blood, and there will be hyperkalemia due to deficiency of insulin. So these are the major changes or metabolic changes which we see in case of diabetic ketoacidosis patient. So that this is the basis. So this is the second part. The first part of diabetic ketoacidosis was already taken by me in the previous video. So I would request you all to go through my previous videos of diabetic ketoacidosis case study, and then you should see my second video that is related to biochemical basis or the reason for the laboratory changes or the medical findings which we see in case of diabetic ketoacidosis. So I request you all to kindly go through my previous videos, or that is case study of diabetic ketoacidosis, and then you should come to this second part where I have explained the basis of the changes which we see in diabetic ketoacidosis. Okay, and remember one thing, the main changes, this ketone bodies can cause severe life-threatening complication, like it can lead to convulsion, it can lead to anxiety, it can lead to various including that can cause cerebral edema also due to that ketone bodies build up inside brain. That can also happen. So all those things you should remember. So thank you all for joining me, and do watch this video.